So I'm going to tell you today about the development of this randomised control trial, but I'm not going to tell you the results. They'll be presented on Wednesday. So my disclosures are that Vitrolife supplied the GX media for the randomised control trial. Reactive oxygen species are essential for cell signalling, but they can also damage cells by peroxidizing and oxidizing lipids, DNA, and proteins. Antioxidants are a wide group of molecules that can protect cells from damage from ROS by deactivating, degrading, and immobilizing them. And when ROS and antioxidants are kept in balance, oxidative stress is kept low. However, when there's an increase in ROS or a depletion of antioxidants, oxidative stress increases, resulting in poor embryo development, aneuploidy, DNA damage, and epigenetic changes. Gametes and embryos can be exposed to a wide range of oxidative stress in vitro, and this can include oxygen, cryopreservation, and centrifugation. Sorry? Louder? Further, multiple exposures to oxidative stress can have a cumulative effect on gametes and embryos. All reproductive fluids contain antioxidants: follicular fluid, seminal fluid, and oviductal fluid. However, these need to be removed for in vitro culture. Gametes and embryos have limited antioxidant capabilities themselves, and sperm in particular are vulnerable to oxidative stress. IVF culture media do contain some molecules with antioxidant properties. However, no culture media have been designed with an antioxidant defense system in mind. And it's been demonstrated that ROS in commercial IVF media can vary between media formulations, and that low levels of ROS in spent embryo culture media are associated with better embryo development and increased pregnancy rates. In animal models, supplementation of individual antioxidants into culture media has been shown to improve outcomes for gametes and embryos by reducing oxidative stress and DNA damage, increasing fertilization and embryo development, and improving cryotolerance. And quite a range of different antioxidants have been tested and shown to be effective. In humans, addition of antioxidants to sperm has been shown to decrease ROS and lipid peroxidation, improve DNA integrity, and increase motility. Addition of antioxidants to immature oocytes as part of IVM has been shown to increase maturation rates, increase spindle morphology, decrease aneuploidy, and increase fertilization and blastocyst development. And finally, one study found that the addition of L-carnitine to human embryos improved embryo quality and clinical pregnancy rates. The effect of antioxidants could vary depending on the culture environment. Since antioxidants should help to ameliorate any oxidative stress that has not already been eliminated by improvements to lab protocols, the benefits observed of adding antioxidants may vary in different settings. This has been demonstrated in animals where it's been shown that the degree of stress in the culture environment decrees how beneficial the antioxidants are. So, for example, with atmospheric oxygen versus physiological oxygen, or individual culture versus group culture. In humans as well, it was shown that adding antioxidants to culture media increased pregnancy rates using a standard incubator, but not using a time-lapse incubator, presumably because of the fewer exposures to atmospheric oxygen in the time-lapse incubator. At the University of Melbourne, our group has been interested in a group of 3 antioxidants: acetyl L-carnitine, which is involved in the transport of fatty acids into the mitochondria, but also prevents and repairs lipid peroxidation; alpha-lipoic acid, which has many functions, including in glucose and lipid metabolism, and is also a regenerator of other antioxidants; as well as N-acetyl L-cysteine, which is a precursor to cysteine, which is involved in the synthesis of glutathione. And this combination of antioxidants has been termed A3. At the University of Melbourne, in David Gardner's group, Mai Trung published a series of papers where she demonstrated that addition of A3 to mouse embryo culture media improved the intracellular glutathione levels, blastocyst cell numbers, and fetal and placental weight. She also found that adding A3 to the IVF insemination media alone decreased hydrogen peroxide in fertilized oocytes and increased blastocyst cell numbers. Also, the addition of A3 to vitrification solutions improved cryosurvival, reduced changes to acetylation, and increased fetal weight. And finally, she found that adding A3 to culture media or vitrification solutions resulted in gene expression in fetal livers and placentas that was more similar to in vivo developed fetuses and placentas. And there were fewer differentially expressed genes with the addition of A3 that were involved in metabolism, cell senescence, and cancer-associated pathways. So all of this evidence from the mouse led to the development of the first sibling oocyte study, which was conducted at the Kuramoto Women's Clinic and the Kiba Park Clinic in Japan. This was a prospective randomized trial on sibling oocytes, where the primary endpoint was good quality blastocysts on day 5. 133 patients were recruited. Patients were under 40, not undergoing PGTA, and had more than 7 oocytes. And the media used was the G-series media from Vitalife, GMOP, GIVF, and G1, G2, with the addition of A3. And this study found that the addition of A3 resulted in more good quality embryos on day 3, but there was no effect on blastocyst rate. While the study was not powered for pregnancy rate, they did observe an increase in the ongoing pregnancy rate for frozen embryo transfers in women aged 35 to 40 years. This then led to the current study, the results of which will be presented on Wednesday, which was a single-centre prospective randomised control trial, where the primary endpoint was clinical pregnancy per patient from the fresh transfer of a single blastocyst. It was determined that 741 patients would be needed per group, and patients were randomised before egg collection. Patients and doctors were blinded to the treatment group, and we used the GX media series from Vitalife, which contains the same A3 combination as our earlier work. The inclusion criteria for this trial were quite broad. There was no age restrictions. The patients just needed to be planning fresh transfer of a single blastocyst. The patient's treatment was conducted according to the clinician's instructions, with the only change being the media used. So in the control group, we used GIVF for gamete collection and insemination, and in the antioxidant group, we used GX-IVF. For embryo culture, we used GTL or GXTL, and embryos were cultured in a time-lapse incubator under 5% oxygen with a refresh of the media on day 5. Single embryos were transferred to the uterus on day 5 under ultrasound, and surplus embryos were vitrified, and pregnancy was assessed by ultrasound. And I can't tell you any more. You'll have to come on Wednesday to session 75. There is a bit of a hint to the outcome of the trial in the title. Antioxidants in culture media improve fertilization rate from ICSI. Thank you.
Rebecca, thank you very much for the lovely talk. Do we have any questions from the audience about the antioxidants in the media? Not at this stage. Rebecca, thank you very much, and thanks to all of you. Please keep an eye on the schedule for the upcoming talks that we have in the studio. Thank you.
Thanks.